Good evening. Welcome to session 3 of um, the interactive sessions of the course Genome Editing and Engineering. This course is taken by Professor Utpal Bora of IIT Guwahati, and I am the TA for this course. I'm from IIT Delhi. So before we get into the questions today, we will be discussing about a brief summary of what was discussed in the week 3. So, can you people see the slides? You can raise your hands. Yeah, okay. So, in week 3, essentially two things were discussed. Homologous recombination and non-homologous recombination and site-specific recombination. Okay, there is an issue of admitting people in. I'll just wait for a minute, try to resolve this. Just a minute. Yeah. Anyway, let's start. So homologous recombination and non-homologous recombination were discussed, and site-specific recombination was discussed. Site-specific recombination is more important in terms of genome editing, as we will be learning in further in the course. So let's see what we have already discussed. So essentially, we discussed about DNA repair mechanisms whenever there is a problem in DNA that could happen due to endogenous factors. That includes replication errors. It could be oxidative damages. It could be a lot of things. So endogenous factors could be there. It could be exogenous factors as well, like UV induced damage and many other things. So whenever such kind of damage happens, the DNA repair machinery comes in play. So we already discussed about base excision repair, where there was DNA glycosylation, creation of a basic site, as you can see here, a basic site, and then this damage is repaired. Another way of damage repair is nucleotide excision repair, that involves whenever there are, let's say, pyrimidine dimers that are created due to UV radiation. In that case, we observe that this kind of damage is repaired using nucleotide excision. So the problematic bases, they are excised out of the DNA, and then they are ligated later, and we get the correct DNA in the end. Another kind of DNA damage repair could be mismatch repair. So we have seen that as well. In this week, we discussed mainly about two other repair mechanisms that involves non-homologous end joining and homologous recombination. Both of this, mainly they work on a specific type of DNA damage, which is called double-stranded breaks. They are referred to as DSBs. So double stranded breaks is any DNA damage. Let's say if the DNA is like this, if it breaks into uh, two DNA fragments, and there is in both the strands, it is damaged. So this kind of damage is called double stranded breaks. So let's see what happens, what are the strategies that can be employed for double-stranded break repair. So two proteins are important. Let's say if in a double-stranded break, if there are involvement of Ku7080 proteins, these are heterodimers, Ku proteins. If these are involved, then the double-stranded break is repaired by the non-homologous end joining. If there is mRNA proteins, mRNA are a group of three proteins, MRE11, RAD50, and NSB1, if I'm not wrong. If three proteins, these are called mRNA proteins. If mRNA protein is involved in a double-stranded break, then usually the repair machinery goes into homologous recombination. So these are major two ways of DNA double-stranded break repairs. Now, what happens in case of homologous recombination is that there are proteins like RAD51 and BRCA2 that facilitate the resection of, firstly, the ends are resected, and then let's say this is the 5' end, this is the 3' end, this is the 3' end, this is the 5' end. So similarly here also we see the same thing. Oh, sorry, 3' end, it should be 5' end, 5' end, and 3' end. So there is a 3' overhang. 3', some extra bases are there. 3' overhang is created, and that is essentially used for strand invasion later. So that 3' overhang, uh, it forms a, what is it called? Complementary base pairing with a homologous strand, which is not damaged. So that is how homologous recombination works. While there are single-stranded annealing also and alternative end joining also that could also happen if homologous recombination and non-homologous end joining do not work properly, then these two mechanisms are involved. So let's see what do we have about homologous recombination. It is known as the guardian of genome integrity because this is one of the most accurate DNA repair mechanisms and most widely used in maintaining the integrity of the genome. So to make sure that your genome is not damaged, homologous recombination is usually the preferred DNA uh, repair machinery, and that is easy in our case specifically because we have homologs, we have two alleles for a particular gene. So that makes it easier to find homologous chromosomes, homologous DNA that is not damaged. So RAT51, it's a structural and functional homologue of bacterial strand exchange protein REC-A, recombinase A. So this homologous recombination process is conserved across various species, right from the bacteria to humans. This process is conserved, and it's a driving force of evolution. So there are three key steps that are involved. First is the strand invasion. This is the first step that happens. Then branch migration happens and new DNA is synthesized. The major polymerase involved here is polymerase delta. And then holiday junction is formed. This structure is called holiday junction, which is further resolved to form an accurate DNA or corrected DNA. During this resolution process, there could be crossing over events or non-crossing over events. So whenever there is crossing over, that means two strands are mixed together. They are joined together. Then we call it as crossing over. In case of non-crossing over, there is no mixing of two strands as such. So talking about, is that clear so far? Maybe a raise of hand would be good. Okay, so if we talk about the mechanism here, DNA ends from double-stranded breaks. They are processed by REC-BCD enzyme complex. So they involve three enzymes together. REC-B, REC-C, and REC-D all have their separate functions. We'll be discussing about it. So they have both nuclease activity and helicase activity. So helicase activity will unword the DNA. It will split open the DNA, and nuclease would start digesting it until a chi site is encountered. Chi site, also written like this, chi site. So this chi site is also known as the recombination hotspot. This is from where recombination occurs. So whenever this chi site is encountered, and it is this chi site is recognized by REC-C, whenever it is encountered, then the nuclease activity is lowered, and REC-A, another enzyme of the recombinase family, REC-A is loaded onto the tail. So we will see this, the function of each enzymes. So first the REC-C, it recognizes the chi site. This is the sequence. It recognizes the chi site, and then it signals REC-D to stop. It signals REC-D to stop. And when stop, REC-D signals REC-B to cut DNA. And after that, there is loading of REC-A. So this is the process, is the flow chart. Now, what could happen if homologous recombination is not possible? If it doesn't happen, then there is another kind of repair process, which is non-homologous end joining. So what happens here, that the double-stranded breaks are recognized by heterodimers. These are proteins, and these proteins, when they bind to the double-stranded break, it allows or it facilitates the docking of DNA PK catalytic subunits. So because of the presence of proteins, they can bind here, and then uh, the damage are processed by tyrosyl DNA phosphodiesterases and other DNA processing enzymes which are involved. And also Artemis, it is also a special protein which repairs any kind of, or it removes any kind of hairpins, any secondary structure of DNA if present, any damaged bases or protein DNA adducts. Then in the end, ligation happens using the complex Lig4, XRCC4 and XLF. Ligation is essentially joining of two DNA strands. And when this ligation happens, um, what happens, what we usually see uh, is the okay, DNA synthesis is not involved, resolution is not involved. These two are not present in non-homologous end joining. So after ligation, the DNA is essentially repaired. So what happens in case of alternate end joining? Just a minute. Alternate end joining acts upon the double-stranded break when already homologous recombination and non-homologous end joining has happened, but they were not successful. So some proteins which are involved in homologous recombination or non-homologous end joining, both of them, they are already present. And they have done some initial processing of the DNA damage, but they were not successful. So hence this alternative end joining happens. It's a distinct repair pathway, and it is always active throughout the cell cycle. However, in the G2 phase, its activity is enhanced. So let's see uh, about lecture 2, we talked about site-specific recombination. So what happens in this case, that the specific site in a chromosome or a genome, they are only recombined. The rest of the genome is not involved in recombination. So that happens. That can be useful for us for targeting a specific, genome alteration in a specific gene, let's say. So what happens is it could be excision where a gene of interest is removed. So these are the recombination spots or recombination points, checkpoints, you could say. So what happens? We will just remove whatever is there in between. So this is called excision. Another thing that could happen is inversion, where the gene of interest is inverted 180 degrees, as you can see here. And what other thing which we could do is insertion. So let's say there is a sequence which is present in a plasmid and it is not present in the genome. So using this process will essentially insert the gene of interest in the genome. So we see this similar kind of strategy in when lambda phage it infects the bacteria, let's say E. coli, then we see this kind of insertion of genes. So we'll talk about it. So site-specific recombination happens when it's a specialized process. It involves reciprocal exchange between two defined DNA sites. It requires two partners, a specific recombinase proteins that is responsible for recognizing the site and for breaking and rejoining the DNA. And it involves a mechanism that involves DNA breakage and reunion with conservation of phosphodiester bond energy. So we will see how that happens. So in case of site-specific recombination, there are essentially two specific recombinases which are involved. One is serine recombinase, another one is tyrosine recombinase. So what happens in serine recombinases? Uh, so you see that it initially the proteins they bind to the double-stranded, about on the target region essentially, and they will cause a 180-degree rotation after they bind. After they bind and cleave, they will cause a 180-degree rotation. So this red part comes down and the blue part goes up. And that is how we see a recombination in this case. So 180-degree rotation is a very important characteristic of serine recombinases. In case of tyrosine recombinases, what we observe is that these are the proteins that the DNA, it binds to the DNA. There is formation of a holiday junction intermediate. The structure is called holiday junction. And later it is then resolved. This holiday junction is resolved and we get recombined DNAs. So in this case, holiday junction intermediate is one of the characteristic features of tyrosine recombinases. So let's see about the questions which we will discuss today. So what is that? What is termed as guardian of genome integrity? Is it cell division, mutation, DNA damage, or homologous recombination? Any answers? Okay, if there are none, then homologous recombination is the answer. We have already looked at it. Uh, DNA ends resulting from a double-stranded break is processed by a multifunctional enzyme complex called REC-BCD, DNA polymerase, RNA polymerase, or gyrase. So whenever there is a double-stranded break, DSB, let's say, this is processed by which enzyme? So the enzyme is REC-BCD. REC-BCD, it is involved in processing double-stranded break first, while DNA polymerase, it is involved in the polymerization process. RNA polymerase is involved for transcription. And gyrase is a type of enzyme. It's one of a type of enzyme called DNA topoisomerases. So it is important for resolving a supercoiled structure of DNA or something like that. So gyrase acts in such a fashion. So a Chi site is a replication hotspot, or DNA damage hotspot, recombination hotspot, or transcription origin. Any answers?
Recombination hotspot.
Yes, you're right. It's recombination hotspot. You're absolutely right. Then in the classical non-homologous end joining pathway, which protein acts as the initial DNA end binding factor? So let's say what happens whenever there is a double strand break, and this has to go, I mean, this double-stranded break has to be repaired using non-homologous end joining. So what will be the proteins that will first interact with this double-stranded breaks? What will be the proteins? Any answer? No answer? Okay, let's go back. Let's go back to a few slides. Let's see what we have already seen. So really sorry for that. Wait. Yeah, so here we see that a double-stranded break, whenever there is Q70 and 80 proteins, they are usually involved in initially for non-homologous end joining repair. So let's go back. So the Ku heterodimers, these are involved for non-homologous end joining repairs. Question 5. Site-specific recombination between sites located on the same chromosome in a head-to-tail orientation. So there is, if this is the recombination sites, and there is some gene inserted here, and if there is the recombination happens that there is only the recombination site, this kind of recombination, or we could say site-specific recombination, is called excision. A tyrosine type recombinase catalyzes formation of holiday junction intermediate. We already said that it's a characteristic of tyrosine type recombinases. The integration of bacteriophage lambda into E. coli is an example of which of these uh, uh, kind of repair, let's say homologous recombination, non-homologous recombination, site-specific recombination, or DNA repair. So the answer to this question is site-specific recombination. So as you can see, whenever in case of lambda phages, there is recombination sites. These are ATTP. And in the same in case of E. coli or in bacteria is ATTB. So whatever is there inside the lambda phage, it is inserted into the specific site. The lambda prophage, it is inserted into the bacterial genome. So this integration happens due to site-specific recombination. Okay, 180-degree rotation and rejoining of cleaved substrate DNA is catalyzed by serine type recombinases or tyrosine type recombinases, both serine and tyrosine type recombinases, or FLP recombinases. So I think we have already discussed this. 180-degree rotation, it's a characteristic of serine type recombinases. So as we see here, that the serine recombinase, it first cleaves the DNA. Then there is a rotation, a 180-degree rotation, which facilitates the exchange. So if you see here from here, to here, there has been a rotation, and this rotation and further ligation forms a recombinant uh, DNA. So that would be all for today. I think I can end the session here.